We're now going to be starting on Chapter 15. This is the special senses. So it's estimated that humans can recognize about 10,000 different odors. Now to make this possible, the nose contains 10 million to even 100 million receptors for the sense of smell, also known as olfaction. And these receptors are contained within a region called the olfactory epithelium. An important note is that smell and taste are both chemical senses. Now we have already talked about this before when we looked at the bones of the skull, but the olfactory epithelium covers the cribriform plate, and that's a part of the ethmoid bone of the skull. And the olfactory epithelium extends along the superior nasal concha. So you can see this olfactory epithelium here in this picture. It has a total area of about 5 centimeters squared. The olfactory epithelium occupies this superior part of the nasal cavity, covering the inferior surface of the cribriform plate of the ethmoid bone and extending along the superior nasal concha. Olfactory epithelium consists of 3 different kinds of cells. These are olfactory receptor cells, the supporting cells, and then also basal cells. Olfactory receptor cells are the first order neurons of the olfactory pathway. So each olfactory receptor, one of these cells, this is a bipolar neuron with an exposed knob-shaped dendrite and also an axon coming up here that extends into the olfactory bulb. Those axons will go through the cribriform plate and end in the olfactory bulb, synapsing with an olfactory bulb neuron, as we can see right here. Now, extending from the dendrite of an olfactory receptor cell are going to be several non-motile olfactory cilia. These are going to be the sites of olfactory transduction. Remember that transduction, this word, is really the conversion of a stimulus energy into a graded potential in a sensory receptor. So in this case, transduction is going to be taking the stimulus energy coming from an odorant molecule and converting that into a graded potential in the olfactory receptor cell. So within the plasma membrane of these olfactory cilia, we have olfactory receptors that detect those inhaled chemicals. And chemicals that have an odor that bind to and stimulate the olfactory receptors in the olfactory cilia are called odorants. We can also call them odorant molecules, but they're often just known as odorants. Oops. So olfactory receptor cells respond to this chemical stimulation of an odorant molecule by producing a generator potential. So this initiates the olfactory response. Kind of initiates how you smell. A couple other cells within the olfactory epithelium, the supporting cells. These are going to be columnar epithelial cells of the mucous membrane lining the nose. So So these cells really provide a lot of physical support. They also provide nourishment and electrical insulation for those olfactory receptor cells. They also help to detoxify chemicals that come into contact with the olfactory epithelium. Basal cells are stem cells located between the bases of supporting cells. Now, these basal stem cells will continually undergo cell division to produce new olfactory receptor cells. The olfactory receptor cells really only live for about a month or so before being replaced. So these new olfactory receptor cells only last for about, I will put about 1 month before they're replaced. Now this process is really quite remarkable considering that the olfactory receptor cells are actually neurons. So like we kind of already learned, mature neurons generally are not really replaced. So olfactory receptor cells are an exception to that rule that neurons aren't generally replaced. Now, within the connective tissue that supports the olfactory epithelium, we have olfactory glands. These are also known as Bowman's glands. And these glands will produce mucus that's carried to the surface of the epithelium by ducts. Now, the secretion is going to moisten the surface of the olfactory epithelium and dissolves odorants within the mucus, and it really helps that transduction can occur here. So that moist surface of the olfactory epithelium is very important. Now, olfaction, just really like all special senses, has a really low threshold. So what this means is that only a few molecules of a certain substance are needed to be present in air in order for you to perceive it as an odor. Adaptation, which is another word for decreased sensitivity or decreasing sensitivity, this adaptation to odors really occurs quite rapidly. So olfactory receptors adapt about 50% in the first second or so after stimulation, but they do adapt kind of slowly after. Still, complete insensitivity to certain strong odors occurs after about a minute after exposure. So you kind of know this because if you go into a room that has a strong odor in it, you notice it right away, but it doesn't take very long for you to be in that room and you don't really notice that odor anymore. This is because of adaptation. So here we have the pathway of olfaction. So the receptors in the nasal mucosa will send branches through the cribriform plate to synapse in the olfactory bulb. And then these impulses will travel along the olfactory tract, and then interpretation of smell is going to occur in the primary olfactory area, which is in the cerebral cortex. On each side of the nose, about 40 bundles of the slender unmyelinated axons of olfactory receptor cells will extend through about 20 of the olfactory foramina in the cribriform plate of the ethmoid bone. So we have these little foramina. We can actually see one right here. It's going to be a little foramina. There's another one here and another one here. So there's about 20 olfactory foramina on each side of the nose that go through the cribriform plate of the ethmoid bone. Now these 40 or so bundles of axons collectively form the right and left olfactory nerve. Now the olfactory nerves will terminate in the brain in paired masses of gray matter called the olfactory bulbs. And these olfactory bulbs are located below the frontal lobes of the cerebrum and lateral to the crista galli of the ethmoid bone. Within the olfactory bulbs, the axon terminals of the olfactory receptor cells will form synapses with the dendrites and cell bodies of olfactory bulb neurons in the olfactory pathway. Now, axons of the olfactory bulb neurons extend posteriorly and they form the olfactory tract, which goes back here. Now, some of the axons of the olfactory tract will project to the primary olfactory area of the cerebral cortex. Now, this primary olfactory area, remember, is located at the inferior and medial surface of the temporal lobe. And this primary olfactory area is where conscious awareness of smell begins. Now, our ability to recognize about 10,000 different odors probably depends on patterns of activity in the brain that arise from activation of many different combinations of olfactory receptor cells. Olfactory sensations are the only sensations that reach the cerebral cortex without first synapsing in the thalamus. So olfactory sensations, they don't synapse in the thalamus. These are going to be our only special sense that doesn't synapse in the thalamus. There are going to be other axons of the olfactory tract that project to the limbic system and also to the hypothalamus. So these projections are going to account for our emotional and memory evoked responses to odor. So once I go to the limbic system and the hypothalamus, these are involved in our emotional response to odors, and also our odor memory. Now, from the primary olfactory area, pathways also extend to the frontal lobe. Now, in a really important region for odor identification and discrimination is the orbital frontal area. So that discrimination of odors allows us to differentiate between different odors. Olfactory receptor cells react to odorant molecules in the same way that most sensory receptors react to their specific stimuli. So a generator potential will develop and trigger one or more nerve impulses. Now, this process, where a generator potential triggers a nerve impulse, in this specific case for olfaction, this process is called olfactory transduction. And so here we have the process of olfactory transduction. So kind of in our step 1 here on the left of this picture, we have the binding of an odorant molecule to an olfactory receptor protein. And this is in the olfactory cilium. Now this is going to stimulate a membrane protein called a G protein. So that's kind of step 1. Odorant molecule binds to the receptor protein, which activates a G protein. We can even label it here, kind of step 1. Okay, so then in step 2, our G protein is in turn going to activate the enzyme adenylate cyclase. And this activation produces a substance called cyclic adenosine monophosphate, otherwise known as cyclic AMP. So that's kind of step 2. G protein will activate adenylate cyclase, which will convert ATP into cyclic AMP. And then our step 3 is where cyclic AMP will open up a sodium channel that allows sodium to enter into the cytosol, which causes a depolarizing generator potential. And it causes a depolarizing generator potential to form in the membrane of the olfactory receptor cell. And this triggers an action potential. So this would be kind of considered step 3 here. So if depolarization reaches threshold, this is when an action potential is generated along the axon of the olfactory receptor cell. So this kind of 3-step process is olfactory transduction. Now moving on to our next special sense, which is taste. Taste, also known as gustation. Just like olfaction, this is also a chemical sense. However, gustation is a lot simpler, much more simpler than olfaction, in that only 5 primary tastes can be distinguished from one another. These are sour, sweet, bitter, salty, and umami. The umami is more of a recently discovered taste than the other 4. It's really kind of described as meaty or savory. Now, all other flavors, things like chocolate or pepper, coffee, these are going to be combinations of the 5 primary tastes. So just like where we had kind of like 10,000 different smells we can distinguish, there's a lot of different tastes we can distinguish, but they're all combinations of our 5 primary tastes. Now the receptors for sensations of tastes are located in the taste buds. Now you have nearly 10,000 taste buds on the tongue, but we also have some found on the soft palate. So remember that soft palate is going to be that posterior portion of the roof of the mouth. We also have taste buds on our pharynx, which is the throat, and even on the epiglottis, which is the cartilage covering our voice box. Something else interesting is that the number of taste buds declines as you get older. Now each taste bud is an oval body consisting of 3 kinds of epithelial cells: supporting cells, gustatory receptor cells, and basal cells. So here on the right we have a single taste bud. You can see the supporting cells here, kind of in the lighter kind of brown pinky color. They contain microvilli and they surround about 50 gustatory receptor cells in each taste bud. Gustatory microvilli, which are gustatory hairs, project from each gustatory receptor cell to the external surface through a taste pore. That taste pore is going to be an opening in the taste bud. And then we have these slightly darker basal cells down here. Again, these are also stem cells found at the periphery of the taste bud near the connective tissue layer down here. And basal cells produce supporting cells, which can then develop into gustatory receptor cells. So one will develop into the other, and that one will develop into these. Now each gustatory cell has a lifespan of only about 10 days. Now this is why it doesn't take the taste receptors on the tongue too long to recover after being burned by a really hot cup of tea or coffee, for example. They replenish themselves rather quickly. Now, taste buds are found in elevations on the tongue that are called papillae. Papillae increase the surface area of the tongue, and they also provide a rough texture to the upper surface of the tongue. Now, 3 types of papillae contain taste buds. There's going to be about 12 very large circular velate papillae. And they form an inverted V-shaped row at the very back of the tongue. Next, we have the fungiform papillae. These are going to be mushroom-shaped elevations that are scattered over the entire surface of the tongue, and they each only contain about 5 taste buds. And then we have the foliate papillae. These are located in small trenches on the lateral margins of the tongue. But most of these taste buds, or most of the taste buds within the foliate papillae, degenerate in early childhood. Now, in addition, the entire surface of the tongue has filiform papillae. Now, these are pointed thread-like structures. They contain tactile receptors, but they do not contain taste buds. So the other 3 papillae are the only ones that contain taste buds. The filiform papillae increase friction between the tongue and food, and they make it a lot easier for the tongue to move food around in the oral cavity. So here we can see those locations of the papillae. Remember that the fungiform and the filiform papillae, they kind of cover the whole surface of the tongue. They're scattered variously, where the velate papillae, these are in that inverted V at the back of the tongue here. And the foliate papillae are located in these small trenches in the lateral margins of the tongue. Three cranial nerves contain axons of the first-order gustatory neurons that innervate the taste buds. These are the facial nerve, which is the 7th cranial nerve. This serves taste buds in the anterior 2/3 of the tongue. the glossopharyngeal nerve, which is the 9th cranial nerve. This serves the posterior 1/3 of the tongue. And then the vagus nerve, the 10th cranial nerve. This serves taste buds in the throat, and the epiglottis. Now here we have the gustatory pathway. The taste buds on the tongue will have nerve impulses propagate along the cranial nerves, the facial nerve, and the glossopharyngeal nerve. And then the taste buds in the throat and epiglottis will travel along the vagus nerve to the gustatory nucleus in the medulla oblongata. Now from the medulla, some axons carrying taste signals will project to the limbic system and the hypothalamus. And others will project to the thalamus. Remember when we talked about olfaction and some of those signals going to the limbic system and hypothalamus that are involved in the emotional response to olfaction and also the memory of olfaction? With the gustatory pathway, it's the same way. So some of those pathways from the gustatory system will go to the limbic system and hypothalamus to involve the emotional response of gustation and also to the memory of gustation, so that you can associate a particular memory, maybe a happy memory out for dinner with family, with a particular taste of a meal, for example. So that will be the emotional response to that taste and also the memory of that taste. Taste signals that project from the thalamus will then go to the primary gustatory area of the cerebral cortex, and this will help to give rise to the conscious perception of taste. Now you may think, if all tastants release a neurotransmitter from many gustatory receptor cells, why do foods taste so different? So the answer to this question is thought to lie in the patterns of nerve impulses in groups of first-order taste neurons that synapse with gustatory receptor cells. So what this means is that different tastes arise from activation of different groups of taste neurons. In addition, although each individual gustatory receptor cell responds to more than one of the 5 primary tastes, it may respond more strongly to some tastants than others. Okay, we're going to be moving on to vision now, the next special sense. So vision, this is the act of seeing. This is really extremely important to human survival. More than half of the sensory receptors in the human body are actually located in the eyes, and a really large part of the cerebral cortex is devoted to processing visual information. Electromagnetic radiation is energy in the form of waves that is radiated from the sun. Now there are many types of electromagnetic radiation, things like this includes gamma rays, X-rays, UV rays, infrared rays, microwaves, even radio waves. These are all types of electromagnetic radiation. Now this range of electromagnetic radiation is known as the electromagnetic spectrum. Now, eyes are responsible for the detection of visible light. Visible light falls within that electromagnetic spectrum, and it has wavelengths ranging from 400 nanometers to 700 nanometers. Visible light exhibits colors, and the color of the light depends on its wavelength. So just for a quick little example, if we have a wavelength here, say that is this long, say this is 400 nanometers long, this would be a violet color. If we have a wavelength this long, for example, this could be 700 nanometers long, this would be a red color. So the color of the light depends on its wavelength. So here we can actually see what the definition of a wavelength is. So the distance between two consecutive peaks of an electromagnetic wave is its wavelength. So just like we kind of saw in the last picture with those two little very basic waves that I drew, you can measure it from the peaks, but you can also just as easily measure it from trough to trough also. It will give you the same measurement. Now wavelengths range from short to long. For example, gamma rays have wavelengths that are smaller than 1 nanometer, and most radio waves have wavelengths that are greater than a meter, which is huge. So let's look at the eyes. So accessory structures of the eye include the eyelids. We have an upper eyelid, which are also called palpebrae. Palpebra is a singular eyelid. Palpebrae is plural eyelids, and a lower eyelid here. Eyelashes are also an accessory structure. Eyebrows. The lacrimal apparatus, which is known as the tear-producing apparatus. It's made up of several different structures, which we will go over a little bit later. And extrinsic eye muscles are also accessory eye structures. So the upper and lower eyelids, the upper and lower palpebrae, these shade the eyes during sleep, and they protect the eyes from excessive light and foreign objects. And the eyelids also help to spread lubricating secretions over the eyeballs to help prevent dry out. Now, as we know, the upper eyelid is going to be a lot more movable than the lower eyelid. And the upper eyelid contains the levator palpebrae superioris muscle. Some other important features surrounding the eyes. The space between the upper and lower eyelids that exposes the eyeball is called the palpebral fissure. The angles of the palpebral fissure, angles on either side, these are known as the lateral commissure, which is narrower and closer to the temporal bone on the lateral side. And then we also have the medial commissure, which is going to be broader and it's nearer to the nasal bone. Within the medial commissure is a small reddish elevation called the lacrimal caruncle, and this contains sebaceous oil glands and sudoriferous or sweat glands. So sebaceous glands and sudoriferous glands. Are within that lacrimal caruncle. The palpebral muscles, like the levator palpebrae superioris, control the movement of the eyelids, whereas the extrinsic eye muscles. These move the eyeball itself so that you can look around. Now, from superficial to deep, we're going to kind of cover the layers of the eyelids. So we'll just put superficial here. So these are going to be the eyelid layers. So the most superficial is the epidermis, then the dermis, subcutaneous tissue, the fibers of the orbicularis oculi muscle, the tarsal plate, which we will talk about right quickly, tarsal glands, and then the conjunctiva, which we will also talk about, but this is the deepest layer of the eyelid. Okay, we'll talk about the conjunctiva, which is right here. This is a really thin protective mucous membrane. It's composed of non-keratinized stratified squamous epithelium with numerous goblet cells. This is really supported by areolar connective tissue. And this is the deepest layer of the eyelid, or below the eyelid. The tarsal plate is this thick fold of connective tissue that gives rise to form and support to the eyelids. Now, embedded in each tarsal plate is a row of elongated modified sebaceous glands that are known as tarsal glands. And these tarsal glands secrete a fluid that helps keep the eyelids from adhering to one another, keeps your eyelids from sticking to each other. Here we have a sagittal section of the eye and the accessory structures, so you can see a better view, or a different view at least, of some of these structures. So let's look at the palpebral conjunctiva. So this lines the inner aspect of the eyelids, and the bulbar conjunctiva passes from the eyelids onto the surface of the eyeball. And this is where it actually covers the sclera. So you can see these here. So here we have the palpebral conjunctiva coming up till here, and then here is going to be the bulbar conjunctiva covering the sclera of the eyeball. The bulbar conjunctiva covers the sclera, but it does not cover the cornea. Now the cornea, this is going to be that transparent region. It forms the outer anterior surface of the eyeball. We'll go over the cornea yet. Okay, let's briefly talk about the eyelashes. They project from the border of each eyelid. And along with the eyebrows, these both kind of help to protect the eyeballs from foreign objects, from perspiration coming off of the forehead, and also protects from the direct rays of the sun. So on this slide, we have the lacrimal apparatus. And we'll see a picture of it in the next slide actually. But the lacrimal apparatus is a group of structures that produces and drains lacrimal fluid, which is also known as tears. And this is all in a process called lacrimation. So the pathway for lacrimation is just down here. So number one, first step in the pathway is the lacrimal glands, then the lacrimal ducts, the lacrimal puncta, the lacrimal canaliculi, the lacrimal sacs, and then the nasolacrimal ducts. That will actually drain the lacrimal fluid into the nasal cavity. So again, lacrimal fluid is just another name for tears, or we commonly call them tears. So here we can see the lacrimal apparatus. A nice picture here, and the nice pathway of the lacrimal fluid. So here we can see these lacrimal glands here. They're each about the size and shape of an almond. And they secrete the lacrimal fluid, which drains into about 6 to 12 lacrimal ducts. And these ducts will empty tears onto the surface of the conjunctiva. So it'll secrete the tears onto the surface of the conjunctiva of the upper eyelid. Now from here, the tears will pass medially over the anterior surface of the eyeball, and they will travel to enter two small openings called the lacrimal puncta. Singular would be lacrimal punctum. The tears will then pass into two ducts, the superior lacrimal canaliculi or canaliculus and the inferior lacrimal canaliculus. Which will then lead into the lacrimal sac and then into the nasolacrimal duct. The duct will carry lacrimal fluid into the nasal cavity, just inferior to the inferior nasal concha. And this is where the lacrimal fluid will mix with mucus. So we can see this pathway here also on the right side. So the lacrimal gland will secrete the tears into lacrimal ducts, which will distribute the tears over the surface of the eyeball. The lacrimal fluid will then enter the superior or inferior lacrimal canaliculi, which will drain the tears into the lacrimal sac on either side of the nose, which will then drain tears into the nasolacrimal duct, draining tears into the nasal cavity. Now, normally tears are cleared away just as quickly as they're produced, either by evaporation or by passing into the lacrimal canals and then into the nasal cavity. The eyes sit in bony depressions of the skull called the orbits, and the orbits help to protect the eyes, to stabilize them in three-dimensional space, and to anchor them to muscles that produce their essential movements. So the orbits in the skull are very important to the eyeball overall. Now there are 6 extrinsic eye muscles that move each eye. They are the superior rectus, inferior rectus, lateral rectus, medial rectus, superior oblique, and inferior oblique. And you'll see this more in the lab material, but these muscles, these extrinsic eye muscles, are supplied by 3 cranial nerves. These are the oculomotor, which is the 3rd cranial nerve, the trochlear, which is the fourth cranial nerve, and also the abducens nerve, which is the sixth cranial nerve. Now the adult eyeball measures about 2.5 centimeters in diameter, and of its total surface area, there's only about a sixth of the eyeball that's exposed, and the remainder is going to be recessed into the orbit, and it's going to be protected by the orbit. Now the eyeball contains two tunics. These are going to be two layers of the retina, or of the eyeball, sorry. These are the fibrous tunic, which is made up of the cornea and sclera, and the vascular tunic, which consists of the choroid, ciliary body, and iris. There is a third wall, or a third layer of the eyeball. This third layer of the eyeball is the retina. So kind of three layers of the wall of the eyeball, the two tunics, fibrous tunic and vascular tunic, and then the retina, which is kind of considered kind of the innermost tunic, some would say. The extrinsic eye muscles extend from the walls of the bony orbit, so the walls of the bone here, and they extend to the sclera, which is the white of the eyeball. So the sclera, we'll see the sclera later, but that's the white part of our eyeball that we see. And so you can see these connections of those extrinsic eye muscles here with the superior rectus muscle, and here for the inferior rectus muscle. These are labeled here. Now the muscles are surrounded in the orbit by a significant quantity of periorbital fat. I don't know if it's labeled here, but I'll just put it in here. So we can see this fat here and here, all surrounding those extrinsic eye muscles. Now these extrinsic eye muscles are capable of moving the eye in almost any direction. Okay, now remember that we have those three layers of the eyeball. The most superficial layer is the fibrous tunic. And this consists of a couple different parts: the anterior cornea, which is labeled here, and also the posterior sclera. So that is labeled here, which is going to be kind of this gray outermost layer of the eyeball here. That will be the sclera. So the cornea first is this transparent coat that covers the colored iris. And because it's curved, as you can see here, this curved cornea, the cornea helps to focus light on the retina. So the retina is the back part of the eyeball here, this yellow internal layer. We'll talk more about the retina a little bit later, but this is the part of the eyeball that allows us to detect light and detect the objects around us. So the cornea helps to focus light onto that retina. Now the sclera, which is also known as kind of just the white of the eye. This is a layer of dense connective tissue made up mostly of collagen fibers and fibroblasts. Now the sclera will cover the entire eyeball except for the cornea, and it has four main roles. So I'll just put the roles of the sclera down here. So roles of the sclera. So the first one is that it gives shape to the eyeball. It also makes the eyeball more rigid. So it's just not a squishy ball. It also protects the inner parts of the eyeball. And it also serves as an attachment site for those extrinsic eye muscles. And we can actually see some of those muscles even here too. The lateral rectus muscle here and the medial rectus muscle here. Two extrinsic eye muscles that attach to the sclera. Now at the junction of the sclera and the cornea is an opening known as the scleral venous sinus, which is also known as the canal of Schlemm. And this is where a fluid called aqueous humor, which we'll describe later, drains into the sinus. So this is a canal where fluid drains through. So just kind of keep that in mind. That's at the corner of the, or at the junction rather, of the cornea and the sclera. So that makes up the fibrous tunic. So let's move on to the second layer of the eyeball, which is the vascular tunic. So the vascular tunic, this is going to be the middle layer of the eyeball, and it actually has three parts. It has the choroid, which is this posterior kind of a pinky layer between the retina and the sclera. The vascular tunic also contains the ciliary body and the iris. So first we'll talk about the choroid. So as you can kind of see in the picture here, it is very highly vascularized, which means there's a lot of blood vessels within the choroid. And this is going to be the posterior portion of the vascular tunic. It lines most of the internal surface of the sclera. Now the numerous blood vessels of the choroid are really going to help to provide nutrients to the posterior surface of the retina. Now in the anterior portion of the vascular tunic, the choroid becomes the ciliary body. Now the ciliary body extends from the ora serrata, which is this little jagged anterior margin of the retina. We can kind of see this here. This little jagged part here is the ora serrata. And the ciliary body extends from there to a point just posterior to the junction of the sclera and the cornea. So right here is where the ciliary body will go to. We'll see a more zoomed in picture of the ciliary body and kind of the anterior margin of the eyeball yet, but we're just going to take this view for now. Now, the ciliary body itself consists of ciliary processes and a ciliary muscle. Now, the ciliary processes are protrusions or folds on the internal surface of the ciliary body, and they contain blood capillaries that secrete aqueous humor. So we're going to secrete aqueous humor, which is a fluid inside the eyeball. We'll talk about the aqueous humor yet, but it is secreted from the ciliary processes. Now, extending from the ciliary processes are zonular fibers. So you can kind of see them right here, these little lines here connecting the ciliary processes to the lens, right here. These zonular fibers are suspensory ligaments that attach directly to the lens. Now, these fibers consist of thin, hollow fibrils that kind of resemble elastic connective tissue fibers. I didn't really talk about this ciliary muscle yet, but the ciliary muscle is a circular band of smooth muscle, and contraction or relaxation of the ciliary muscle changes the tightness of the zonular fibers, and it's going to alter the shape of the lens. So this ciliary muscle changes the shape of the lens by contracting or relaxing. And it changes the tension on those zonular fibers that are connected to the lens. So changing the shape of the lens adapts it for near vision or for far vision. So the ciliary muscle is a really important muscle inside the eyeball. The iris is the colored portion of the eyeball, and essentially it's shaped like a flattened donut. Now, it's suspended between the cornea and the lens, and it's attached at its outer margin to the ciliary processes. Now, the principal function of the iris is to regulate the amount of light entering the eyeball through the pupil, which is the hole in the center of the iris. So again, the principal function of the iris is to regulate the amount of light entering the eyeball through the pupil. Now, it's really going to be autonomic reflexes that regulate the pupil diameter in response to light levels. So the first one we have here is in bright light circumstances. So when bright light stimulates the eye, the parasympathetic fibers of the oculomotor nerve are going to stimulate the circular muscles of the iris to contract. So we have these circular muscles that go all the way around the pupil. And when they contract, they make the pupil smaller, which is constriction of the pupil. Now in dim light, over here, this is where we have sympathetic neurons will stimulate radial muscles to contract. So these are going to be these muscles. So when they contract, the radial muscles themselves get smaller, which causes the pupil itself to get larger, resulting in dilation. An increase in pupil size is dilation, and a decrease in pupil size is constriction. Constriction allows less light into the eyeball, whereas dilation allows more light into the eyeball. The third and innermost layer of the eyeball, the retina, lines the posterior 3/4 of the eyeball, and it's the beginning of the visual pathway. Now, through a device called an ophthalmoscope, which shines light into the eye, an observer can view a magnified image of the retina. You can see the associated blood vessels, and you can also see the optic nerve. Now, the optic disc is the site where the optic nerve exits the eyeball. Now, the optic disc is also called the blind spot, and this is because it contains no photoreceptors in that area. Couple of other points of the retina we want to talk about. The macula lutea. This is in the exact center of the posterior portion of the retina. This is going to be the visual axis of the eye, or the part of the retina that gives you the highest acuity. Light that hits the macula lutea will give you the best focus. And the fovea centralis, this is a small depression in the very center of the macula lutea. This contains only cones, and this area is going to be the area of the highest visual acuity. So here we can see the retina. This is seen through an ophthalmoscope. Now, the surface of the retina is really the only place in the body where blood vessels can actually be viewed directly and examined for pathological changes. So this is where you would see changes that occur in diabetes mellitus, changes in cataracts, and other age-related macular diseases. So here we can see the optic disc, where the optic nerve leaves the retina. And bundled together with the optic nerve is the central retinal artery, a branch of the ophthalmic artery, and the central retinal vein. So you can kind of see these blood vessels around the optic disc there. Also visible here is the macula lutea and the fovea centralis. Now, the retina contains photoreceptors. Now, these are the sensory receptors that detect light. These are rods and cones. Now, rods allow us to see in dim light, but they don't detect color. Essentially, they're like grayscale photoreceptors. Cones, on the other hand, detect color. Now, the retina contains a couple of different layers. There is a pigmented layer and a neural layer. The pigmented layer is a sheet of melanin-containing epithelial cells that are located between the choroid and the neural part of the retina. So the choroid is actually going to be this layer here. Just adjacent to that pigmented layer. And this pigmented layer contains melanin. Now the melanin in the pigmented layer of the retina helps to absorb stray lights. Now the choroid also kind of acts in this way to help absorb stray lights, but the melanin really acts a lot in that. Now the neural layer, this is the sensory layer of the retina, and it is a multi-layered outgrowth of the brain that processes visual data extensively before sending nerve impulses into axons that form the optic nerve. Now there are three distinct layers of retinal neurons: the photoreceptor cell layer, the bipolar cell layer, and the ganglion cell layer. So these three layers are all contain retinal neurons. These layers are separated by two zones: the outer synaptic layer and the inner synaptic layer. These layers are going to be where synaptic contacts are made. So this is where our synapses are between adjacent neurons. Now this part is really important. The eyeball is going to be shaped like this, kind of around here, with the lens and the cornea over here, right? So your eye is facing this direction. So light is actually going to pass through the ganglion cell. Light is going to come this way. So light comes this way, and it's going to pass through the ganglion cell layer. It's going to pass through the bipolar cell layer and both synaptic layers before it even reaches the photoreceptor layer. So light is going to come all the way back through all these layers of neurons. It's going to activate these rods and cones. And then the nerve impulse is going to travel in the opposite direction to the incoming light. So that's going to be the direction of the nerve impulse from that light. So it's kind of confusing, but just know that the light passes through all the layers of neurons in the retina before it hits those photoreceptors, and then the nerve impulse that light creates is going to travel in the opposite direction. So we can see in this picture, we can see the rods that look very rod-like, cylindrical, and we can also see the cones that look very cone-like. We can also see these bipolar cells. These are these long blue cells there. Those are going to be the bipolar cells. And we can also see these orange ganglion cells. Now, as you kind of notice, there's also two other types of cells that are present in the bipolar cell layer of the retina. These are horizontal cells, which is this kind of octopus-looking cell here, and an amacrine cell, which is this little cell down here. Now these cells form laterally directed neural circuits. So let us say lateral neural circuits. And essentially, these modify the signals being transmitted along the pathway from photoreceptors to bipolar cells to ganglion cells. So they modify the signal. Now, photoreceptors are specialized cells that convert light rays into nerve impulses. The rods, like I said, allow us to see in dim light, but they don't produce any color vision. It's only going to be the cones that produce color vision. Now, there's 3 types of cones. There's blue cones, which are sensitive to blue light. Green cones are sensitive to green light. And red cones, you may have guessed it, but they are sensitive to red light. Now really it's going to be the stimulation of the combination of these 3 types of cones that allows us to view the world in this rainbow of colors that we see. Now from the photoreceptors that we can actually see in this picture right here, you can see them here. There's kind of this layer of our photoreceptors on both sides here. The information is going to flow through the outer synaptic layer to the bipolar cells, and then from bipolar cells through the inner synaptic layer to ganglion cells. And then down here we have those ganglion cells, and their axons will extend, make it maybe purple here, their axons are going to extend posteriorly to the optic disc, which is right labeled here. So we're going to have these axons of the ganglion cells extend to the optic disc and then exit the eyeball as the optic nerve. Now, as you can see, we have photoreceptor cells that kind of end here and end here. So we have this entire section from here to here that doesn't have any rods or cones. So you can kind of see how the optic disc is going to also be called a blind spot. There's no rods or cones here. So any light that comes in and hits right here is actually going to hit nothing. It's not going to hit any rods or cones, and it's not going to be detected just because there's no photoreceptors in this area. So it's a blind spot. We can't see any images that strike this part of the retina. Behind the pupil and the iris, within the cavity of the eyeball, is the lens. Now the lens divides the interior of the eyeball into 2 cavities. The anterior cavity, which is the space anterior to the lens, consisting of 2 chambers, and the posterior cavity. Now the 2 chambers of the anterior cavity are the anterior chamber and the posterior chamber. The anterior chamber lies between the cornea and the iris, and the posterior chamber lies behind the iris, but in front of the zonular fibers and the lens. Both chambers of the anterior cavity are filled with aqueous humor, which is a clear, watery fluid. Now, the larger posterior cavity of the eyeball is also called the vitreous chamber. This lies between the lens and the retina. Now, within the vitreous chamber is the vitreous body, which is also called vitreous humor. This is a transparent jelly-like substance that essentially holds the retina flush against the choroid, which gives the retina an even surface for the reception of clear images. So if we kind of just give a brief example of the eyeball here with the cornea at the front. Really bad drawing of an eyeball, but that's the cornea at the front. Okay, so here we have a normal retina kind of filling the posterior end of the eyeball, right? And we have this jelly that fills the whole back of the whole posterior cavity. We have this vitreous humor, which is this jelly that pushes against the retina and holds it in place against the back of the eyeball. Now, without the vitreous humor, the retina may be like this, kind of filling the interior of the eyeball. And with this, we would not get a very clear image at all when light would hit the retina. It'd be very wavy and mismatched. I don't know what it would look like, but it wouldn't be clear at all. So having this vitreous humor, this transparent but gelatinous substance holding the retina against the posterior, holding it against the choroid, giving it a really flush surface, we really get a clear reception of images. So light will pass through the cornea, then the anterior chamber, then to the pupil, then the posterior chamber, which is the posterior chamber of the anterior cavity. Then light will pass through the lens, the vitreous humor, and then is projected onto the retina. Now remember that aqueous humor fills both chambers of the anterior cavity. So here we have the anterior cavity here, the anterior chamber in front of the iris, that posterior chamber being behind the iris. Now, aqueous humor continually filters out of blood capillaries in the ciliary processes of the ciliary body, and it enters into the posterior chamber. So here we're going to get that aqueous humor flow here. Aqueous humor is then going to flow forward between the iris and the lens here, through the pupil, and then into the anterior chamber. Now it could go either way in the anterior chamber, really anywhere all around the anterior chamber, but from the anterior chamber, the aqueous humor is going to drain into the scleral venous sinus. Remember the canal of Schlemm? So it's going to drain here into that little sinus there, and then back into the blood. So this is kind of the flow or the cycle of aqueous humor. So again, this is now normally aqueous humor is completely replaced about every 90 seconds. And this is because we just have this continual flow of aqueous humor all the time. So here is a summary table of the structures of the eyeball. So this is a really, really helpful table because it gives you a picture of where the structures are located and their function. So here we have two tables of structures, or two columns, or two, and then we have two columns of their function. So We have the fibrous tunic containing the cornea and sclera, the vascular tunic with the iris, choroid, and ciliary body, and then the retina. We also have the lens, the anterior cavity, and the vitreous chamber. Now first, to understand how the eye forms clear images of objects on the retina, we have to examine three processes. So our first process that we have to look at is the refraction or bending of light by the lens and cornea. We also have to look at accommodation. Accommodation is the change in shape of the lens, which we will look at yet. And then we also have to look at the constriction or narrowing of the pupil. So these three processes will help us to get a better understanding of how the eye forms clear images of objects on the retina. So first off, we're going to look at refraction. So when light rays travel through a transparent substance, such as air, and they pass into a second transparent substance with a different density, like water, they bend at the junction between the two substances. Now this bending is called refraction. So we can see this refraction here, and that's because it's the change in density between these two mediums. Because if we kind of kept this path going, our light rays would have ended up there, but instead they bend at the junction between those two densities of mediums. Now, an interesting thing is that images focused on the retina are inverted or upside down and right to left reversed. So that is that the light from the right side of an object strikes the left side of the retina and vice versa. Now the brain corrects this and actually interprets them as the right way around. Now there's about 7% of total refraction of light occurs at the cornea. The lens provides remaining 25% of focusing power, and it also changes the focus to view near or distant objects. Now when an object is 20 feet away or more from the viewer, the light rays reflected from the object are nearly parallel to one another. So the lens has to bend these parallel rays just enough so that they fall exactly focused on the central fovea. Now this is where the vision is going to be the sharpest. Now because light rays that are reflected from objects closer than 20 feet are divergent rather than parallel, the rays have to be refracted more if they're to be focused on the retina. So this additional refraction is accomplished through a process called accommodation, and this is completed by the lens. So accommodation occurs if the object is closer than 20 feet from the viewer. So first we have to kind of know the difference between concave and convex. So this would be a convex lens, whereas this would be a concave lens. So a surface that curves outwards, like the surface of a ball, is said to be convex. So if we draw kind of a lens here, we can kind of actually draw kind of like this, those curved outward. Now, when the surface of a lens is convex, that lens will refract incoming light rays towards each other so that they'll eventually intersect. So say, for example, we have light rays coming in this way, and here, when they hit this surface, they're going to be refracted inward. So they kind of follow the glass here. So they're going to continue going until they intersect. Now, on the other hand, if the surface of a lens curves inward, like a hollow ball, for example, the lens is said to be concave. It also kind of looks like a cave entrance. And concave lenses will cause light rays to refract away from each other. So again, if we have light rays coming in here, they come here and they follow the glass, so they're going to go like this. So that's just kind of basically how convex and concave lenses work, how they refract light that hits them. So here we have kind of different ways that the eye can look at objects. Now, when the eye is focusing on a really close object, so say for example right here, we have this little pin here, I guess, that's really close, the lens has to become more curved, which causes a greater refraction of the light rays. And this is because the light rays from a close object are diverged. And so if they were to continue on their path, they wouldn't even come close to the fovea centralis here on the eyeball. They will barely even touch the retina just because these diverging rays diverge a lot. So in order to kind of counteract that, the lens has to curve a lot more. So as it becomes more curved, kind of gets more rounded, and this extra curve in the lens causes a greater refraction of the light rays. So this turns into a stronger convex surface and brings those light rays together. So you can see like that. Now, this increase in the curvature of the lens for near vision is called accommodation. Now, the near point of vision, this is a term that we give to the minimum distance from the eye that an object can be clearly focused with maximum accommodation. Sorry, so this is called the near point of vision. So again, this is the minimum distance from the eye that an object can be clearly focused on with maximum accommodation. Now, for a young adult, this distance is approximately 4 inches. So how does accommodation occur? When you're viewing distant objects, the ciliary muscle of the ciliary body is relaxed. And the lens is going to be flatter, and this is because it is stretched in all directions by really tight zonular fibers. So it kind of sounds counterintuitive, but when the muscle is relaxed, the zonular fibers are tight. Now, when you view a close object, the ciliary muscle contracts. And this pulls the ciliary processes and the choroid forward toward the lens. This action actually releases the tension on the lens and the zonular fibers. Now, because it's elastic, the lens is elastic, the lens actually becomes more spherical when the ciliary muscle contracts. So when the lens becomes more spherical, it becomes more convex, which increases the focusing power and causes that greater convergence of the light rays. Remember, it's going to be the oculomotor nerve that innervates the ciliary muscle of the ciliary body, and it mediates the process of accommodation. It's going to be the parasympathetic fibers of the oculomotor nerve. So again, when you see a distant object like the top picture here, the ciliary muscle is relaxed and the zonular fibers are tight, making the lens flatter. When you look at a very close object, the ciliary muscle contracts, zonular fibers become relaxed, and it makes the lens more convex. The normal eye, known as an emmetropic eye, this can sufficiently refract light rays from an object 20 feet away so that a clear image is focused on the retina. This means that the light rays coming in are going to converge directly onto the retina so that they cross paths right at that point on the retina. Many people do lack this ability because of refraction abnormalities, though. Now, among these abnormalities is myopia. This is also known as nearsightedness, and this occurs when the eyeball is too long to the focusing power of the cornea and lens, or also sometimes happens when the lens is thicker than normal. And what this means is that the image is going to converge in front of the retina. So it says right here, image converges in front of the retina. We'll see that in the next slide. Now, myopic individuals can see close objects clearly, but they can't see distant objects very well. A concave lens is going to be used to correct this type of vision. So here we can see a myopic eye, and we can see that those light rays will converge at a point in front of the retina here. Now, most errors in vision can be corrected by eyeglasses, contact lenses, or by surgical procedures. For this minor myopia, we can see that it can be corrected with the use of a concave lens. So this is because in the myopic eye, the image focus point is in front of the retina, and the concave lens changes the focus point to be further back so that the light rays will actually converge directly on the retina, further back than they normally would. In hyperopia, or farsightedness, the eyeball length is short compared to the focusing power of the cornea and lens. Or sometimes the lens is thinner than normal. Both of these cases result in an image that converges behind the retina. Hyperopic individuals can see distant objects very clearly, but can't see close objects very clearly. A convex lens is used to correct this type of abnormality. So here we can see that hyperopic eye. We can see the retina is right here, but we can see that those light rays converge at a point behind the retina. So in this case, we have a convex lens that is used to correct this hyperopic eye. This type of lens is going to diverge the entering light rays so that the focus point comes forward to be directly on the retina, as we can see here. Another refraction abnormality is astigmatism. And this is where either the cornea or the lens has an irregular curvature. Now, as a result, parts of the image are out of focus, and so the vision is going to be blurred or distorted. Rods and cones are named for the different appearance of the outer segment. So the outer segment is labeled here. The rods are on the left. They have a rod-like structured outer segment. The cone on the right has a cone-like shaped outer segment. So the outer segment is going to be that distal end next to the pigmented layer. Transduction of light energy into a receptor potential occurs in the outer segment of both rods and cones. So the outer segment is where the transduction actually occurs. Photoproteins are integral proteins in the plasma membrane of the outer segment. In cones, the plasma membrane is folded back and forth, kind of in a pleated fashion. In rods, on the other hand, the pleats pinch off from the plasma membrane and they form discs. The outer segment of each rod contains a stack of about 1,000 discs that are piled up like coins inside of a wrapper. Photoreceptor outer segments are renewed at a really astonishing rapid pace. In the inner segment down here, this contains the cell nucleus, the Golgi complex, and many mitochondria. You can see in both the rods and the cones there is a ton of mitochondria. Now the first step in visual transduction is the absorption of light by a photopigment. Photopigment is a colored protein that undergoes structural changes when it absorbs light, and it's found in the outer segment of a photoreceptor. Now, light absorption initiates the events that lead to the production of a receptor potential. Now, a single type of photopigment in rods is called rhodopsin. Now, there are 3 different cone photopigments that are present in the retina, one in each of the 3 types of cones. Color vision results from the different colors of light selectively activating the different cone photopigments. We're really just going to be looking at rhodopsin though, since it will simplify it a little bit for us. All photopigments associated with vision contain 2 parts. There is a glycoprotein known as opsin. Opsin is a glycoprotein. And there's also, within the photopigments, a derivative of vitamin A that is called retinol. Now, photopigments respond to light in a cyclical process. We're going to see that in the next slide here. So here we have that cyclical process. Here we can see the opsin with the retinol inside. Together they make up rhodopsin. So let's start off here with our step number 1 on the right here, isomerization. Now in darkness, retinal has a bent shape, so it's called cis-retinal. Cis-retinal is bent and it fits very snugly into the opsin portion of the photopigment. Now when cis-retinal absorbs a photon of light, it straightens out into a shape that is called transretinal. This cis to trans retinal conversion, this is called isomerization, and this is the first step in visual transduction. Our next step is number 2. It's called bleaching. So in about a minute, transretinal completely separates from opsin. The final product looks pretty colorless, so this part of the cycle is termed bleaching of the photopigment. Because as you can see here, when it has cis-retinal, it is a colored photopigment, and when it isomerizes to transretinal and leaves opsin, the opsin becomes colorless. So it's kind of a bleaching process. We have step 3 here. This step is the retinal isomerase step. So an enzyme called retinal isomerase converts the transretinal back into cisretinal. So that is an entire step here. And then our 4th step in this cycle is the regeneration step. So this is where the cisretinal can then bind to opsin again, and it reforms that functional photopigment. This part of the cycle is the resynthesis of the photopigment, so it's called regeneration. When you emerge from dark surroundings into sunshine, this is when light adaptation occurs. So your visual system adjusts in seconds to the much brighter environment, and it does so by decreasing its sensitivity. On the other hand, when you enter a darkened room, your visual system undergoes dark adaptation. This takes several more minutes as sensitivity increases quite slowly over time. Now the difference is the rate of bleaching and regeneration of the photopigments in the rods and cones. And this accounts for some of the sensitivity changes during light and dark adaptation. The absorption of light and isomerization of retinal initiates chemical changes in the photoreceptor outer segment that leads to production of a receptor potential. In darkness, sodium ions flow into the photoreceptor outer segments through ligand-gated sodium channels. It's going to be these blue channel here. Now the ligand that holds these channels open is cyclic guanosine monophosphate, or cyclic GMP. So this is the ligand that holds these ligand-gated sodium channels open. Now the inflow of sodium, this is called the dark current, and this partially depolarizes the photoreceptor. So in darkness, cyclic GMP holds those sodium channels open, and sodium goes into the outer segment of the photoreceptor, and it partially depolarizes the photoreceptor. Now, as a result, in darkness, the membrane potential of a photoreceptor is about -30 millivolts. Now this is much closer to zero than a typical neuron's resting membrane potential of negative 70 millivolts. Now the partial depolarization during darkness triggers this continual release of the neurotransmitter at synaptic terminals. The neurotransmitter in rods and perhaps in cones is the amino acid glutamate. Now at synapses between rods and some bipolar cells, glutamate is an inhibitor neurotransmitter. So what glutamate does as it is coming out at these synapses is that it triggers those inhibitory postsynaptic potentials. It triggers IPSPs, inhibitory postsynaptic potentials. And IPSPs, remember, will hyperpolarize the postsynaptic cell. So it's going to hyperpolarize the bipolar cells. And the glutamate will actually prevent bipolar cells from transmitting signals to the ganglion cells. So we have this here. Glutamate released at these synaptic terminals will inhibit the bipolar cells. So essentially, in darkness, the photoreceptors are partially depolarized, and so they're kind of on the on state because they are pumping out glutamate down here. And when the photoreceptors are on, glutamate inhibits bipolar cells, and so it turns off kind of the rest of the pathway. Bipolar cells are off, so they're not sending any signal on, so on and so forth. So in darkness, the whole pathway is turned off because the photoreceptors are kind of turned on. Now when light strikes the retina and cis-retinol undergoes isomerization, enzymes are activated to break down cyclic GMP. So now we no longer have that cyclic GMP to activate those ligand-gated sodium channels. So as a result, some of those cyclic GMP-gated sodium channels will close, and the sodium inflow decreases, and the membrane potential becomes more negative. So it approaches negative 70 millivolts. Essentially, it hyperpolarizes the cell. So this sequence of events produces a hyperpolarizing receptor potential that decreases the release of glutamate. So glutamate release is turned off, and so it's no longer inhibiting the bipolar cell, and the bipolar cell is going to be excited and is going to send out action potentials. So the takeaway from this is that light excites the bipolar cells that synapse with rods by turning off the release of an inhibitory transmitter. The excited bipolar cells will then subsequently stimulate the ganglion cells to form action potentials in their axons, and essentially this is how we see. Visual signals in the retina undergo considerable processing at synapses among the various types of neurons in the retina. Remember, we have horizontal cells, bipolar cells, and amacrine cells. Then the axons of the retinal ganglion cells provide output from the retina to the brain, exiting the eyeball as the optic nerve. Now the pathway from the retina to the brain is here. So as the optic nerve leaves the eyeball, goes to the optic chiasm. Remember, this is where the optic nerves cross over. Then to the optic tract. Then to the lateral geniculate nucleus of the thalamus. And then there's optic radiations that bring the information to the primary visual areas in the occipital lobes of the brain. This is where perception is received. So here we can see those optic nerves. Those axons within the optic nerve will pass through the optic chiasm. It's a crossover, as in the letter X, so they kind of form the letter X right here at that optic chiasm. This is going to be the crossing point of the optic nerves, and some axons will cross to the opposite side, but others remain uncrossed. After passing through the optic chiasm, the axons are now part of the optic tract, and they enter the brain, and some of them will terminate in the lateral geniculate nucleus of the thalamus. And here, they will actually synapse with neurons whose axons form the optic radiations. Kind of these large fanning out projections. These are the optic radiations here. And the optic radiations will project to the primary visual areas of the cerebral cortex. Remember, that's in the occipital lobe. And this is where visual perception begins. Now, some of the fibers in the optic tracts will terminate in the superior colliculi. And these are going to be ones that control extrinsic eye muscles. And also, some of the fibers in the optic tracts will also terminate in the pretectal nuclei. And these control the pupillary and accommodation reflexes. Now, everything that can be seen by one eye is that eye's visual field. Now, because our eyes are located anteriorly on our heads, the visual fields actually overlap quite considerably. Now, we have binocular vision due to the large region where the visual fields of the two eyes overlap. And this overlapping portion is called the binocular visual field. So we can see here that the orange is going to go to the left eye. So this portion here is going to go to this part of the left eye. The red is going to go to this part of the left eye. And then we also have the right eye, where we have this lighter orange color goes to the right side of the right eye here. That's going to be this portion. And then we have the yellow goes to the left side of the right eye there. Now, the visual field of each eye is divided into two regions. We have the nasal side, or the central or medial, and the temporal side. which is the lateral half. For each eye, light rays from an object in the nasal half of the visual field will fall on the temporal half of the retina, and the light rays from an object in the temporal half of the visual field fall on the nasal half of the retina. So visual information from the right half of each visual field is conveyed to the left side of the brain, and visual information from the left side or left half of each visual field is conveyed to the right side of the brain. We'll see that in the next slide, kind of the logistics of it here. So first off, we can see here for each visual field of each eye that the nasal half of the visual field travels to the temporal side of the retina. You can see that for both the left and the right eye. And we also see that the temporal half of the visual field travels to the nasal half of the retina on both sides. Now the axons of all the retinal ganglion cells in one eye exit the eyeball at the optic disc, and then they form the optic nerve on that side. So all of these retinal ganglion cells are going to form the optic nerve for the left side, and all these cells are going to form the optic nerve on the right side. So that would be number 1 here, the optic nerve. Now at number 2 is the optic chiasm. And at the optic chiasm, axons from the temporal half of each retina do not cross, but they continue directly back to the lateral geniculate nucleus of the thalamus. And that's going to be on the same side. So we could write this out here. It's going to be the axons coming from the nasal side of the retina that do cross over. So you can see those nasal retina axons cross over here and also here at that optic chiasm. So the nasal half of each retina cross at the optic chiasm, and then they continue to the opposite side of the thalamus. So when we get to the optic tract, behind or posterior to the optic chiasm, the optic tract is going to consist of crossed and uncrossed axons that project from the optic chiasm to the thalamus on one side. Now, axon branches of the retinal ganglion cells will project to the midbrain on both sides here. And this is where they participate in neural circuits that govern constriction of the pupils in response to light. And it also works in coordination of head and eye movements. So the axons that go here are involved in that. Constriction of pupils. Remember, that is in response to light, and also coordination of head and eye movements. Now these branches will also extend to the suprachiasmatic nucleus of the hypothalamus. And this establishes patterns of sleep and other activities that occur in a circadian or daily schedule. And this is in response to the intervals of light and darkness. So we can, well, actually we could write that out too. So the branches also go to the suprachiasmatic nucleus of the hypothalamus. And this establishes pattern of sleep. And essentially other circadian activity schedules, or other activities on a circadian schedule, we should say. So this is in response to light and dark cycles. Now the axons of the thalamic neurons will form optic radiations, and this is as they project from the thalamus to the primary visual area of the cortex on the same side. So we're going to be moving on to hearing and equilibrium. So hearing is the ability to perceive sounds. The ear is really an engineering marvel because its sensory receptors can transduce sound vibrations with amplitudes as small as the diameter of an atom of gold into electrical signals that are 1,000 times faster than the photoreceptors respond to light. So it's really quite amazing. Now the ear also contains receptors for equilibrium. So remember, equilibrium is the sense that helps you to maintain your balance and also to be aware of your orientation in space. Now the ear is divided into 3 regions: the external ear, the middle ear, and internal ear. Now the external ear consists of the auricle, the external auditory canal, and the eardrum, which is also known as the tympanic membrane. The auricle is going to capture the sound, and the external auditory canal transmits that sound to the eardrum. Now next to the exterior opening, the auditory canal contains a few hairs and specialized sweat glands, which are called ceruminous glands. And they secrete cerumen, which is also known as earwax. Now this combination of hairs and cerumen helps to prevent dust and foreign objects from entering the ear. Cerumen also prevents damage to the delicate skin of the external ear canal by water and even insects. Okay, so the auricle, this is a flap of elastic cartilage that's kind of shaped like the flared end of a trumpet, and it is covered by skin. Now the rim of the auricle is called the helix, and the inferior portion is called the lobule. Ligaments and muscles will attach the auricle to the head. You can even see the elastic cartilage here that makes up that structure of the auricle. The external auditory canal, this is a curved tube about 2.5 centimeters long that lies in the temporal bone, and it leads to the eardrum. Here we have the tympanic membrane right here, also known as the eardrum. This is a really thin and kind of semi-transparent partition between the external auditory canal and the middle ear. The tympanic membrane is covered by epidermis and is made up of simple cuboidal epithelium as well. Now, between the epithelial layers is a connective tissue composed of collagen, elastic fibers, and fibroblasts. And tearing of this tympanic membrane is called a perforated eardrum. Oops. It's kind of when you blow your eardrum, it kind of breaks that membrane, that tympanic membrane. The middle ear is a small air-filled cavity in the petrous portion of the temporal bone that's lined with epithelium. Now, the middle ear is separated from the external ear by the tympanic membrane, and it's separated from the internal ear by a thin bony partition that contains two small openings, the oval window and the round window. Now, extending across the middle ear and attached to it by ligaments are the three smallest bones in the body. These are the auditory ossicles. The auditory ossicles are connected to each other by synovial joints. Now, named for their shapes, they are the malleus, incus, and stapes. Commonly, they're known as the hammer, anvil, and stirrup, respectively. Now, the anterior wall of the middle ear contains an opening that leads directly into the auditory tube, or this is also the pharyngeotympanic tube, also known as the eustachian tube. Now, this tube acts to regulate the air pressure in the middle ear. So here we can see kind of a close-up of the middle ear. We can see the handle part of the malleus attaches to the internal surface of the tympanic membrane. Now, the head of the malleus articulates with the body of the incus. The incus, which is the middle bone in the series, articulates with the head of the stapes. Now, the base, or kind of the footplate part of the stapes, fits into the oval window. Now, directly below the oval window is another opening. This one's not actually covered, and this is the round window. The round window is enclosed by a membrane called the secondary tympanic membrane. Now, besides the ligaments, there's two tiny skeletal muscles that also attach to the ossicles. First is the tensor tympani muscle. This is supplied by the mandibular branch of the trigeminal nerve. And this tensor tympani muscle really limits movement of the bones, and it also increases tension on the eardrum to prevent damage to the inner ear from really loud noises. So prevent damage from those loud noises. Kind of the two main roles of that tensor tympani muscle. We also have the stapedius muscle, which is labeled down here. This one is supplied by the facial nerve. So remember, this is the 7th cranial nerve. And this is the smallest skeletal muscle in the entire body. Now, by dampening large vibrations of the stapes due to loud noises, it protects the oval window. But it also decreases the sensitivity of hearing. So dampens large vibrations. So dampening those large vibrations of the stapes due to loud noises, it protects the oval window there. Now moving on to the internal ear, which is also called the labyrinth. And it's called this because it's a really complicated series of channels. Now structurally, it consists of two main divisions. First is the outer bony labyrinth. And the outer bony labyrinth is going to enclose an inner membranous labyrinth. Now the bony labyrinth is a series of cavities in the petrous portion of the temporal bone, and it's divided into three areas. The semicircular canals, the vestibule, and the cochlea. Now the bony labyrinth, we can see is labeled here, is lined with periosteum, which we would assume would line bone, and it contains a fluid called perilymph. So this is important here, this fluid. So this fluid, which is chemically similar to cerebrospinal fluid, surrounds the membranous labyrinth. Now the membranous labyrinth is a series of epithelial sacs and tubes inside the bony labyrinth that generally has the same form as the bony labyrinth, and the membranous labyrinth houses the receptors for hearing and equilibrium. The membranous labyrinth contains a different type of fluid, which is called endolymph. Okay, here we can see the vestibule. This is the oval central portion of the bony labyrinth. Now the membranous labyrinth in the vestibule contains two sacs, which are called the utricle and the saccule. So the bony labyrinth is the vestibule, and then the membranous labyrinth inside the vestibule is the utricle and saccule. And these two are connected by a small duct. Now projecting superiorly and posteriorly from the vestibule are three bony semicircular canals. Now each of which lies approximately at right angles to the other two. So they kind of lie at like an X, Y, and Z axis kind of thing. This is kind of how those semicircular canals are oriented. Now based on their positions, they're named the anterior, the posterior, and lateral semicircular canals. Now the cochlear duct, this is a continuation of the membranous labyrinth into the cochlea. Now it is filled with endolymph. The channel above the cochlear duct is the scala vestibuli, which ends at the oval window. The channel below is the scala tympani, which ends at the round window. Both the scala vestibuli and the scala tympani are part of the bony labyrinth of the cochlea. Therefore, both of these chambers are going to be filled with perilymph. So here we can see the cochlea. We can also see its relation to the utricle and saccule and to these semicircular canals. So at one end of each semicircular canal is a swollen enlargement called the ampulla. And these portions of the membranous labyrinth that lie inside the bony semicircular canals are called semicircular ducts. So it's kind of like we have this part, which is the semicircular canal. And then this part inside is the semicircular duct. So again, the bony labyrinth is going to be filled with perilymph, whereas the membranous labyrinth, the semicircular duct, is going to be filled with endolymph. Now these semicircular ducts connect with the utricle of the vestibule. Now just anterior to the vestibule is the cochlea. This is it's a bony spiral canal that kind of resembles a snail's shell. So we can see this here. So we can see that internal cochlear duct. We can also see the scala tympani and scala vestibuli that we just kind of talked about in that last slide. So here we have that cochlear duct inside and the scala vestibuli and tympani just outside of that. Now the vestibular branch of the vestibular cochlear nerve consists of ampullary, utricle, and saccular nerves. Now these nerves contain both first order sensory neurons and motor neurons that synapse with the receptors for equilibrium. The cochlear branch of the vestibular cochlear nerve connects to the spiral ganglia that connects to the organ of Corti within the cochlear duct. So the cell bodies of the cochlear branch of the vestibular cochlear nerve are found within the spiral ganglia, just like the cell bodies of the sensory neurons for equilibrium are found in the vestibular ganglia. Now the vestibular membrane separates the cochlear duct from the scala vestibuli, and the basilar membrane separates the cochlear duct from the scala tympani. Now resting on this basilar membrane is the spiral organ, also known as the organ of Corti. So here we have that organ of Corti. This is a coiled sheet of epithelial cells, including supporting cells and about 16,000 hair cells, which are going to be the receptors for hearing. So hair cells in the inner ear are the receptors for hearing. Now there are two groups of hair cells. These are the inner hair cells, which are arranged in a single row, and the outer hair cells, which are arranged in 3 rows. At the apical tip of each hair cell, there are several stereocilia and 1 kinocilium that extend into the endolymph of the cochlear duct. So this part kind of up here above the tectorial membrane, this is the cochlear duct. Now, as sound waves move the fluid in the cochlear duct, it will move the tectorial membrane, and the hair cells attached to the tectorial membrane will also move. Now, because these hair cells are mechanical receptors, this movement will initiate a nerve impulse. Kind of go over that just a little bit more in just a second. Now, at their basal ends, the inner and outer hair cells will synapse both with the first order sensory neurons and with motor neurons from the cochlear branch of the vestibular cochlear nerve. Remember that those cell bodies of the sensory neurons are located in the spiral ganglion. So it's just going to be kind of like down here somewhere. So that's where the cell bodies are found of these sensory neurons. Okay, so this tectorial membrane, this is a flexible gelatinous membrane up here. Oh, this membrane up here. And it covers the hair cells in the spiral organ. The stereocilia of the hair cells are going to be embedded within this tectorial membrane, while the bodies of the hair cells rest on the basilar membrane. You can see that basilar membrane down here. And then down here we have the scala tympani. So this area is the scala tympani down here. Just kind of getting oriented how this works in larger space. So here we have the events that are involved in hearing. So at number 1, we have the auricle directing sound waves into the external auditory canal. Now, when the sound waves strike the tympanic membrane here, the alternating waves of high and low pressure in the air cause the tympanic membrane to vibrate back and forth. So the tympanic membrane vibrates slowly in response to low frequency or low pitched sounds. It's going to be slow vibrations. And the tympanic membrane is going to vibrate very rapidly in response to high frequency or high pitched sounds. Now the central area of the tympanic membrane, remember, connects to the malleus, which vibrates along with the tympanic membrane. This vibration is transmitted from the malleus to the incus and then to the stapes. Now as the stapes moves back and forth along with the tympanic membrane, its oval-shaped foot plate, which is attached by the ligament to the circumference of the oval window, is also going to vibrate within that oval window. The vibrations at the oval window are about 20 times more vigorous than those of the tympanic membrane, and this is because the auditory ossicles will efficiently transmit small vibrations that are spread over the large surface area of the tympanic membrane into larger vibrations at a smaller surface, being the oval window. So large surface area of the tympanic membrane. If we have small vibrations here, this is going to be kind of transmitted into larger vibrations on a small surface area of the oval window. And this is transmitted really efficiently by the auditory ossicles. So those are the malleus, incus, and stapes. Now the movement of the stapes at the oval window sets up fluid pressure waves in the perilymph of the cochlea. As the oval window bulges inward into the cochlea, it pushes on the perilymph of the scala vestibuli. So pressure waves will travel along the scala vestibuli all the way around, and they're going to be transmitted from the scala vestibuli to the scala tympani as they kind of travel around the helicotrema, and they go all the way around the scala tympani, and eventually they're going to make it to the round window. So that round window being right here. Now, as those pressure waves get to the round window, it causes it to bulge outward into the middle ear. Now, the pressure waves will travel through the perilymph of the scala vestibuli, kind of going back a little bit, then at the vestibular membrane, and then they're going to be moving into the endolymph inside the cochlear duct. So you can kind of see they kind of go interior as well into that interior cochlear duct. Now the pressure waves in the endolymph of the cochlear duct cause the basilar membrane to vibrate, which moves the hair cells of the spiral organ against the tectorial membrane. Now this leads to the bending of the stereocilia and ultimately to the generation of nerve impulses in those first order neurons in the cochlear nerve fibers. Now sound waves of various frequencies cause certain regions of the basilar membrane to vibrate more intensely than other regions. So because of this, each segment of the basilar membrane is tuned for a particular pitch. So if the vibrations occur more intensely here at the beginning of the cochlear duct, this is going to be more for high frequency, or it's going to be detected more as high frequency. But if it occurs more here, close to the helicotrema, this will be more tuned for low frequency pitch. We'll see that in the next slide here. Now, because the membrane is narrower and stiffer at the base of the cochlea, so here is going to be narrower and stiffer here, high frequency or high pitched sounds will induce the maximal vibrations in this area. toward the apex of the cochlea, kind of more out here. The basilar membrane is wider and more flexible. And so we're going to get low frequency or low pitched sounds that will cause more maximal vibration of the basilar membrane here. Loudness is determined by the intensity of the sound waves. So high intensity sound waves will cause larger vibrations in the basilar membrane, which leads to a higher frequency of nerve impulses reaching the brain. Now, louder sounds also may stimulate a larger number of hair cells. The bending of the stereocilia of the hair cells in the spiral organ, or the organ of Corti, causes the release of the neurotransmitter glutamate. And glutamate is going to generate a nerve impulse in the sensory neurons that innervate those hair cells. So these are going to be the hair cells of the inner ear. Now remember, those cell bodies of the sensory neurons of the organ of Corti are located in the spiral ganglia. Now nerve impulses will pass along the axons of these neurons, which form the cochlear branch of the vestibular cochlear nerve. Now these axons will synapse with neurons in the cochlear nuclei in the medulla oblongata on the same side. These impulses will then travel to the medial geniculate nucleus in the thalamus, and they're going to end in the primary auditory area of the cerebral cortex. Now, some of the axons from the cochlear nuclei will decussate in the medulla. So we can kind of see, you can see this decussation here. And they decussate in a tract called the lateral lemniscus to the opposite side, and then they're going to terminate in the inferior colliculus in the midbrain. Now, other axons from the cochlear nuclei will end in the superior olivary nucleus in the pons, and this will be on both sides of the pons. Now, there will be slight differences in the timing of nerve impulses arriving from the two ears at the superior olivary nuclei, and this slight difference of timing allows us to locate the source of a sound. If it's more on our right side, the nerve impulse will come to the right side of our pons quicker than to the left side. Now, axons from the superior olivary nuclei will also ascend in the lateral lemniscus tracts on both sides, and they're going to end in the inferior colliculi. Now, from each inferior colliculus, the nerve impulses are conveyed to the medial geniculate nucleus in the thalamus. And then finally, they will travel to the primary auditory area in the cerebral cortex. So we can see this whole pathway of hearing here. Now there are two types of equilibrium or balance. There is static equilibrium. This refers to the maintenance of body position, so mainly the head, but it really is that maintenance of the position of the body relative to the force of gravity. So most commonly when you're in a car that is speeding up or slowing down, static equilibrium will help you to maintain your head position. The other type of equilibrium is dynamic equilibrium, and this is the maintenance of body position in response to sudden movements like rotational acceleration or deceleration. These are going to be the main ones. Collectively, the receptor organs for equilibrium are called the vestibular apparatus. Now, all of the receptor organs within the vestibular apparatus include the saccule, the utricle, and the semicircular canals, or the semicircular ducts, rather. Now, the walls of both the utricle and saccule contain small thickened region called a macula. And the two maculae are perpendicular to one another. These are going to be the receptors for static equilibrium. So the receptors for static equilibrium are found within the utricle and saccule. And the receptors for dynamic equilibrium are going to be the semicircular ducts. Now the maculae consists of two kinds of cells, the hair cells, which are the sensory cells, sensory receptor cells, and also supporting cells. The hair cells on their surface will have 40 to 80 stereocilia of graduating height plus one kinocilium. As in the cochlea, the stereocilia are going to be connected by tip links. So if we I quickly kind of draw a hair cell here. So just imagine this is a hair cell, the nuclei. This is going to be a sensory receptor right here. And so our hair cells are going to be like this, and with one larger kinocilium here. Now they're actually connected all with tip links, these little connection that holds the tips of all of them together. So this green part is going to be tip links, and this is just so that all of the hair cells kind of travel together, or all the stereocilia travel together, sorry. So that if they all move in one direction, they all go this way, then the tip lengths will help them to kind of move together as a group. Now collectively, the stereocilia and the kinocilium are called a hair bundle. Now scattered among the hair cells are columnar supporting cells that secrete this really thick gelatinous glycoprotein layer, which is called the otolithic membrane. So the otolithic membrane will actually rest on top of the hair cells. It's kind of like we have this otolithic membrane like this. Now movement of the head causes gravity to move the otolithic membrane down over the hair cells. And remember, these hair cells will synapse with the first order sensory neurons in the vestibular branch of the vestibular cochlear nerve. We'll see a better picture here on the next slide of this poorly drawn picture that I've drawn. Okay, so here we can see that otolithic membrane. We can see those hair cells with the hair bundles coming out of them, And those hair bundles, the stereocilia and the kinocilium, protrude into that otolithic membrane. And there's a layer of dense calcium carbonate crystals that are called otoliths that extend over the entire surface of the otolithic membrane. Now, because the otolithic membrane sits on top of the macula, if you tilt your head forward, the otolithic membrane, along with the otoliths, is going to be pulled by gravity. So it slides downhill over the hair cells in the direction of the tilt, pretty much bending all the hair bundles along with it. However, if you are sitting upright in a car and the car suddenly jerks forward, the otolithic membrane lags behind the head movement. It's going to pull on the hair bundles and makes them bend in the other direction. Now, bending the hair bundles in one direction stretches the tip links, which pulls open the transduction channels, producing depolarizing receptor potentials. So tip links, not only will they hold the hair bundles together, but they are also connected to the transduction channels. So they're going to open those mechanically gated channels. Now, bending in the opposite direction will close the transduction channels and produce hyperpolarization. So here we can see a little bit closer even those hair cells with the stereocilia and kinocilium. So we can see how they're embedded within that otolithic membrane with the otoliths on top. Now, as the hair cells depolarize and repolarize, they release neurotransmitter at faster and slower rates. So remember that these hair cells will synapse with these first order sensory neurons. So right here, it's going to be that Remember, these are going to be first order sensory neurons of the vestibular branch of the vestibular cochlear nerve. Now, these neurons will fire impulses at a slow or rapid pace, depending on the amount of neurotransmitter present. There's also going to be motor neurons that synapse with the hair cells. These motor neurons will help to regulate the sensitivity of the hair cells and even the sensory neurons. The three semicircular canals contain the semicircular ducts, and they function in dynamic equilibrium. Now, the ducts lie at right angles to one another in three planes. The two vertical ducts are the anterior and posterior semicircular ducts, and then the horizontal one is the lateral semicircular duct. Now, this positioning of these semicircular ducts permits the detection of rotational acceleration or deceleration. Now, there's an ampulla in each duct that contains crista with a group of hair cells. Now, movement of the head affects the endolymph and directly affects the hair cells. Now, this is going to generate a receptor potential leading to nerve impulses. So here we can see that crista within the base of the semicircular duct. So we can kind of see on the left here what we're actually looking at, this ampulla in the base of the semicircular duct. So this is a dilated portion of each duct, and it has this small elevation called the crista. And each crista contains a group of hair cells and supporting cells. Now, covering the crista is a mass of gelatinous material called the cupula. It acts very similar to the otolithic membrane. So when you move your head, the attached semicircular ducts and hair cells move with it. The endolymph within the ampulla, however, is not attached, and so it's going to lag behind. So as the moving hair cells drag along the stationary endolymph, the hair bundles will bend. So you can kind of see this here with the head rotating. The cupula is dragged through the endolymph, and then the hair cells are bent in the opposite direction. Now, the bending of the hair bundles produces receptor potentials. In turn, the receptor potentials will lead to nerve impulses that pass along the vestibular branch of the vestibular cochlear nerve. The bending of hair bundles of hair cells in the semicircular ducts, utricle, or saccule causes the release of the neurotransmitter glutamate, which generates nerve impulses in the sensory neurons that innervate those hair cells. So glutamate is also the neurotransmitter that we're going to see for equilibrium. Now, the cell bodies of sensory neurons are located in the vestibular ganglion here. And the nerve impulses will pass along the axons of these neurons, which form the vestibular branch of the vestibular cochlear nerve. Now, most of these axons will synapse with sensory neurons in the vestibular nuclei, which is the major integrating center for equilibrium in the medulla oblongata and the pons. So I guess it's not really labeled here, but we'll quickly put here that this is in the medulla oblongata and in the pons there. Now the remaining axons will enter the cerebellum through the inferior cerebellar peduncles. And there's going to be bidirectional pathways that connect the cerebellum and the vestibular nuclei. Now, the vestibular nuclei integrate information from the vestibular, visual, and somatic receptors, and then it's going to send commands to 4 different areas. So this is from the vestibular nuclei, where information is going to from there. So our first one is to the nuclei of cranial nerves. So we have the oculomotor nucleus, the trochlear nucleus, and the abducens nerve nucleus that control coupled movements of the eyes with those of the head to help maintain focus on the visual field. So that's going to be the first areas where these neurons will travel to. Second one could be the nuclei of the accessory nerve. So this one specifically helps to control head and neck movements to assist in maintaining equilibrium. So that one's going to be just down here. The third place that this information may go would be to the vestibulospinal tract. So this conveys impulses down the spinal cord to maintain muscle tone in skeletal muscle to help maintain equilibrium. And the last place they may go would be to the vestibular posterior nucleus, the ventral. Here we go. Ventral posterior nucleus, excuse me. And this is the nucleus in the thalamus, which is labeled here, ventral posterior nucleus in the thalamus. And then from there, it will go to the vestibular area in the cerebral cortex. And this is actually part of the primary somatosensory area. Now the next couple slides are just going to be summarizing tables that are really helpful for studying, but essentially just goes over everything that we've gone over so far. So here, this table is going to summarize the structure of the ear relating to hearing and equilibrium. So in this particular slide, we have the external ear with their parts and the function. Here is the middle ear with the ossicles and the auditory tube, and then their functions as well. And here is the inner ear with the cochlea and the parts of the vestibular apparatus. Remember, the vestibular apparatus includes the semicircular ducts, the utricle, and the saccule. So this one's quite important. Now, most people don't experience any problems with the senses of smell and taste until about the age of 50. And this is due to a gradual loss of olfactory receptor cells and gustatory receptor cells, coupled with their slower rate of replacement as we age. Now in the eyes, there's going to be several age-related changes that can occur. Now the lenses begin to lose some of their elasticity, and thus they can't change their shape easily. So this results in a condition called presbyopia, which is essentially just difficulty on focusing on close objects. Muscles of the iris also begin to weaken and will react more slowly to light and dark, which causes elderly people to have difficulty adjusting to changes in lighting. Now, some diseases of the retina are more likely to occur in old age, including age-related macular disease and glaucoma. So, age-related macular disease, this is also called AMD sometimes. This is a degenerative disorder of the retina in people usually 50 years old and older. Abnormalities occur in the region of the macula lutea, which is ordinarily the area of the most acute vision. But these patients retain their peripheral vision, but they lose the ability to see straight ahead. Glaucoma. This is the most common cause of blindness in the US. Usually it affects about 2% of the population over 40. Now this is due to an abnormally high intraocular pressure as a result of a buildup of aqueous humor within the anterior cavity. Now generally it's quite painless and the other eye compensates largely, so a person may experience considerable retinal damage and loss of vision before they're even diagnosed. Now by about the age of 60, about 25% of individuals experience a noticeable hearing loss, especially for higher pitched sounds. Now the age-related progressive loss of hearing in both ears is called presbycusis. Now it may be related to damaged and lost hair cells in the spiral organ or degeneration of the nerve pathway for hearing. Tinnitus, sometimes pronounced tinnitus, this is ringing in the ears. Vestibular imbalance also, these both can occur more frequently in the elderly. This slide isn't really going to be tested on too much, more just for interest's sake.